Hello and namaste. I'm Dr. Sonam Chaudhary. I work as an assistant professor in the Department of Clinical Physiology. We are here today to discuss about the physiology question which appeared in Common Entrance Examination 2077. So let's begin. The first question which appeared in last year was, which of the following is true about bile salt? The options were stimulates its own production, stimulates bilirubin secretion, contracts gallbladder. The option D is observed from the jejunum. So here you can see that among this option, stimulates bilirubin secretion. Okay, this bilirubin, as we all know, it is being formed from the lysis of RBC. So it has got no such relation with the bile salt. So you can easily omit this option. So there is now confusion between these three, A, C, and D. To know about the correct answer, let us recall some facts about the bile salt. So we know that the bile salt is responsible for the digestion of the fats. One of the very important functions of the bile salt is it is helping in emulsification of fat. That means it is breaking the larger fat droplets into the smaller one. And when the fat droplets are converted into the smaller fat droplets, the total surface area of the fat droplets increases so that the lipase enzymes can act more over it and the digestion of the lipids can occur. Similarly, the bile salt is also the one which is forming the micelles, and via this micelle formation, they are transporting these fat globules along the intestinal epithelial cell line, and it is helping in the absorption of the fats as well. And as we know, the bile salt is helping in the fat digestion, and it is also interesting to know that the one which is helping fat digestion is being formed from the fat only, that is from the cholesterol. The cholesterol is the precursor of the bile salt, from which this cholesterol is actually being converted into primary bile acid in the liver, and the sodium and potassium salt or the primary bile acids are actually what we call the bile salt. Okay. Now, these primary bile acids are secreted along the bile, and then they are released into the intestine. In the intestine, these bile salts, they are acted upon by the intestinal bacteria, and then they are converted into the secondary bile acid. And it is also important to know that most of this bile salt, which is being secreted in this intestine, around 90 to 95% of this bile salt, they are again reabsorbed back into our circulation. And this is very important. This enterohepatic circulation of bile salt is very important. The enterohepatic circulation is very important because due to the availability of the bile salt in the stool only, the digestion of the meal, whatever we are taking in a day, that is being possible. Okay. So let us move into the regulation of the secretion of this bile. So there is the neural regulation and there is the humoral regulation. So whenever we are talking about the neural regulation, remember that since we are talking about digestion and absorption, the parasympathetic nerves, they are playing very important role. Now in this parasympathetic nerves also, we know that these are the vagal efferents. The vagal efferents, they are going to release the neurotransmitter like nitric oxide and VIP in this junction of the sphincter oda, which is going to cause the relaxation of this area, due to which the secretion of the bile into the second part of the duodenum is possible. Similarly, the bile, if it is not being secreted, right, they are being stored in the gallbladder. We all know this thing. And for the secretion of the bile from the gallbladder also, the gallbladder has to be contracted. And for the contraction of the gallbladder, it is again the acetylcholine, which is being released from the vagal efferents. It is playing very important role for the contraction of the smooth muscle of this gallbladder. Okay. Similarly, the cholecystokinin, which is a GIT hormone, it is also playing important role for the contraction of the gallbladder. So now with this point, what you can see now, if you are talking about is bile salt causing the contraction of the gallbladder? No, because we have already come to know that the contraction of the gallbladder is being done by the acetylcholine and cholecystokinin. Is it being reabsorbed from the jejunum? No, because the bile salt, they are being reabsorbed from the terminal ilium. And remember that this reabsorption process is also because of the secondary active transport, that is sodium biosalt co-transporter is working over there for the reabsorption, for the absorption of this bile salt. Okay, so the correct answer would be the bile salt is stimulating its own production. So option A. is our right answer. And this kind of substances which is causing the stimulation of its own secretion or production or stimulation of production of the bile, we also call this kind of substances as cholinergic substance. Okay, now let's move into our second question. Okay, so let's move into our next question. What is true about swing and stance phases in gait cycle? The options given were: stance phase occupies 40% and swing phase occupies 60%. The stance phase occupies 70% and the swing phase occupies 30%. The stance phase occupying 60% and the swing phase 40%. The option D was: the stance phase occupies 30% and the swing phase occupies 70%. So now to know about the correct answer, firstly we have to know about the gait cycle. So we all know that the gait is actually the style or the manner of our walking. So whenever we are walking, there's the movement of both of our upper limbs as well as lower limb. And the movement of lower limb is playing very important role because it is actually propelling our body forward. And that's what the intention of the movement is, right? And similarly, the lower limb is also the one which is balancing our body. So whenever our lower limb is moving, it's not like both of our legs will move together. One leg will move forward and the other leg will balance the body. Okay. So whenever the leg is moving forward and it is in the air, we call it the swing phase. And whenever the leg is in the ground and supporting our body, we call it the stance phase. And it is very important to know that the balancing of our body is also very important while we are walking, otherwise we may fall down. So eventually, whenever we are tracing the movement of a particular limb, we found that the stance phase, that means whenever the foot is in the ground, that phase is actually occupying 60%, and the swing phase, that is when the foot is in the air, that is occupying 40%. Okay, so that means that our movement is basically trying not only to move forward, but we are also balancing our body simultaneously. So this would be our correct answer. The correct answer is C. Here you can see that the stance phase occupies 60% and the swing phase occupies 40% of our gait cycle. Okay. Now, our third question is, which of the following parameter is used to regulate parathyroid hormone? The options given were total calcium, ionized calcium, potassium, and phosphate. Now, the parathyroid hormone, we all know that it is responsible for the regulation of calcium homeostasis in our body, right? So, let us recall some brief the function about the parathyroid hormone. So, this hormone is actually being produced from the parathyroid gland. We all know this thing, right? And they are having the primary effect in three important sites: the bones, the kidneys, and the intestine. And remember that the action of the parathyroid hormone in the bone and in the kidneys, they are the direct action. That means whenever the parathyroid hormone is acting in the bone, they are increasing the osteolytic activity, osteoclastic activity, so as to increase the level of calcium in our blood. Similarly, the action of parathyroid hormone in the kidneys is causing more reabsorption of the calcium from the kidney tubules, due to which, again, it is helping in the collection. It is helping in the restoring the calcium level in our blood. And in the intestine, the parathyroid hormone is acting indirectly. That means via the activation of this vitamin D. Okay, and via this activated vitamin D, it will eventually help in the absorption of calcium present in our diet. Now, the calcium, which is the ultimate thing which is being regulated from the parathyroid hormone, is total plasma level is 10 milligram per deciliter. And in this 10 milligram per deciliter, they can be in the diffusible form or they can be bounded with the protein which we call the non-diffusible form, right? Now, in the diffusible form also, 4.72 milligram per deciliter of the calcium, they are in the ionized form. And remember that this ionized calcium or this free calcium is the one which is giving the stimuli for the production of the parathyroid hormone from the parathyroid gland. So obviously, whenever the calcium level goes down, the parathyroid hormone has to be produced so as to conserve or increase the calcium level in our body, right? So you can see that the parathyroid hormone, which is being produced from the chief cell of the parathyroid gland. These chief cells, they are having certain receptors which are the calcium sensing receptors, and these receptors are actually going to detect the decrease in the free ionized calcium level of the blood. Okay, and whenever they are going to detect this thing, that the calcium level is down in the blood, then this receptor will be activated. And remember that these calcium sensing receptors, they are the G protein coupled receptors. And via this GQ subunit, they are going to cause the phosphoinositide crossover. Okay. And eventually there will be so many processes inside the cell and leading to the increase in the secretion of the parathyroid hormone into the blood, which is eventually having its action in the bone, kidney, and indirect action in the gut. So now with this overall uh, thing about the parathyroid hormone, we can easily solve this question that the one which is going to regulate the parathyroid hormone level in the blood is the ionized calcium. The option B, it will be our right answer. Okay, let us move into our fourth question. Which of the following is true about insulin metabolism? The options given were increased intake of glucose adipocyte, increased gluconeogenesis. The option D was decreased glycolysis, and then increased uptake of glucose in hepatocyte. Now, whenever we are talking about insulin, we all know this thing that insulin is responsible to decrease our blood glucose level. And from this option only, one of the options which you can easily omit is the option B, that is increased gluconeogenesis. Why? Because we know that the gluconeogenesis is the process in which there will be the formation of new glucose molecule from the non-carbohydrate source. And if there's a formation of the glucose molecule, they are eventually going to cause the increase in blood glucose level. So that is not the action of the insulin. So here, the option B can be easily omitted. The remaining options are now increased intake of glucose in adipocyte, glycolysis, and about uptake of glucose in hepatocyte. And to know about this thing, let us know about the function of insulin very briefly. So this insulin is being produced from the beta cells of the pancreas and then released into the blood, right? The pancreas also have this dual function that they are also secreting the pancreatic juice. Okay. When we are focusing in the insulin, that is the endocrine function of the pancreas, the insulin, now we are going to focus in its role in the glucose only. Okay. So they have found that in the rapid action, within second, the insulin is leading to the increased transport of glucose into the insulin sensitive cell. Okay, that means this action is very, very rapid. That means they are going to, the insulin whenever is there in the blood, they are going to cause the transport of glucose into the insulin sensitive cells. Now within minutes, the insulin will lead to the activation of the glycolytic enzymes, due to which there will be increased glycolysis, there will be increased utilization of the glucose. And whenever there is increased utilization of the glucose, the level of the glucose inside the cell will decrease, and this is actually facilitating the transport of the glucose in some way. Similarly, they also going to activate this glycogen synthase, due to which now the glucose would be converted into a storable form, that is glycogen, right? So via this action, that means by the activation of the glycolytic enzymes and glycogen synthase, the both of this process, they are actually facilitating the transport of glucose from outside the blood or outside the cell into the cell. But we also know that the glucose is a water soluble substance, water soluble molecule. Okay, so they cannot easily cross the cell membrane. For crossing the cell membrane, obviously they require some transporter, and the transporter which is playing role for the transport of the glucose can be the second co-transporter like sodium glucose co-transporter, or the carriers like the GLUT transporter. The GLUT, they have been divided into 7 types. You can see in this picture, in the slide, from GLUT 1 to 7. So now among all of these transporter, the one which is dependent upon insulin is GLUT4. Okay, that means the transport of the glucose via this GLUT4 transporter is dependent upon insulin, and this GLUT4 transporter, they are found in the skeletal, cardiac muscle, adipose tissues. Okay, so this primary sites you have to understand, you have to memorize. So the GLUT4 is present in this area, skeletal, cardiac, and adipose tissue site. Okay, so now with this we can make out which one of the options is right. So if you're talking about the insulin, its role, is it causing decreased glycolysis? No. Similarly, is it causing increased uptake of glucose in hepatocytes? No. Okay, because the uptake of glucose into the hepatocytes is actually being done by the activity of this glucokinase enzyme inside this hepatocytes, due to which there will be the transport of glucose into the cell in of the liver. Okay, so the correct answer would be option A, that is increased intake of glucose in the adipocyte, because this adipocyte, they are having this GLUT4 transporter which is dependent in insulin. So we'll now move into our next question. After the resection of the ilium, the patient presents with loss of joint, vibration and position sensation. Deficiency of which vitamin may lead to these symptoms? The options were vitamin A, vitamin B1, vitamin B2 and vitamin B9. Okay, so now with this question, we have to know about the different clinical feature which appear with the deficiency of this vitamin. So whenever we are talking about vitamin A or retinol, right, we all know that vitamin is playing very important role in our vision, right? So obviously, the deficiency of the vitamin A would have problem in the eyes, the dry eyes, the night blindness. These are the common thing. Also, the dry skin. And whenever we are talking about vitamin B2 deficiency or riboflavin deficiency, then these kind of deficiency would lead basically the problems around our mouth, in the lips, okay, in the corner of the mouth, ulcers, ciliosis, glossitis, this kind of features you can see. The basic thing. There are many other features also. So this point you have to remember. Okay, this the swollen corner of the mouth, a cracked mouth, the mouth ulcers. These are the things which you can see. And whenever we are talking about vitamin B9 or folic acid, this deficiency is leading to the anemia, because folic acid is very much important in the erythropoiesis process. So whenever B9 is deficient in the body, the immature RBC will be produced and released into the blood, which are bigger in structure. So this bigger RBC are prone to destruction and may even really lead into the anemia. So this kind of anemia, we also call it the macrocytic anemia, right? So whenever there's anemia, there's the symptoms like irritability, fatigue, there will be loss of appetite, and also in the vitamin B9 deficiency, the tongue swelling. Okay, this kind of problems, the anemia related problems you can see. Now, in case of vitamin B1 deficiency, which is a thiamine, this thiamine is actually playing very important role in the citric acid cycle, which is actually a common pathway for the generation of energy from many macronutrients in our body, right? So the vitamin B1 deficiency would eventually lead to the energy depletion in the body, which will lead to the symptoms like fatigue, depression, memory impairment. And if we are going to categorize its features, these can be broadly categorized into also one which is going to affect our nervous system, okay, and the other one which is going to affect our cardiovascular system. And affecting the cardiovascular system is actually a more life threatening one, and this kind of condition we also call it wet beriberi. And if they are going to affect our nervous system, then you call this condition dry beriberi. They have found that this thiamine is actually playing very important role for the formation of the myelin sheath, okay, for the proper functioning of these neurons as well, formation of the neuron as well. So whenever the thiamine deficiency is there in the body, and if it is of dry beriberi type, okay, then the neurological symptoms can happen, like there will be loss of tendon reflexes, or they can be the burning or tingling sensation, numbness of the feet. This all neural symptoms can also appear in the body. So with this now we can say that vitamin A, retinol would related with the eye and skin problem. Vitamin B2 is related with the problem around our mouth. Vitamin B9 is related with the anemic like features. So eventually the correct answer would be vitamin B1, and it will lead to the neurological and mental disturbances as we have just discussed. So after the resection of the ilium, the vitamin deficiency is actually leading to this symptom in the patient. Now, okay, let's move into our next question. All can give an inaccurate reading with pulse oximeter except jaundice, methemoglobinemia, henna, nail paint. So here we all know the function of the pulse oximeter. It is basically going to detect the oxygen saturation of our blood. And whenever we are talking about oxygen saturation of the blood, it is like in the blood also, the oxygen is basically being transported with the heme molecule, with the hemoglobin molecule present inside the RBC, right? So whenever the hemoglobin is transporting the oxygen, remember that the iron which is present in the hemoglobin, that is in the ferrous form. And this ferrous form of the iron is actually binding the oxygen molecule very loosely. Okay, so the oxygen itself is not being utilized, and the ferrous form of the iron is not being oxidized to form the ferric form in the normal condition. But if that happens congenitally or by the acquired condition, you call this condition methemoglobinemia. So in the methemoglobinemia, the ferrous form, instead of the ferrous form of the iron, there is a ferric form of the iron because they have been oxidized. So here you can see that in this condition, the oxygen is being utilized, and although this oxidized iron can now no more transport the oxygen, and the body will eventually suffer from the hypoxia and cyanosis. The other features can happen in the body, but the pulse oximeter would show the saturation of around 80 to 85%, or in very severe condition, they can show the saturation of 60 to 65%. Okay, that means since the oxygen level is there in the blood, okay, so they would actually give the inaccurate reading. Similarly, if you are looking at the technical aspect of this pulse oximeter, so this pulse oximeter is a device which is used to measure the oxygen level of the blood and heart rate, oxygen level of the blood. Okay, so here they are going to detect how much oxygen is present in the blood based on the infrared and red light which passes through the finger. Okay, so whenever we are having this nail paint or henna at this condition also, although the oxygen level in the blood is right or normal, but the passes of this infrared or red light can be obstructed, okay, due to which the level of the oxygen may be, oxygen saturation which is showing may be wrong. Okay, so now with this we can conclude that the methemoglobinemia is a wrong option. Henna and nail paint, they are also wrong option, and the correct answer would be jaundice. That means the jaundice, the condition in which there will be increase in the serum bilirubin level, right? In this condition, the pulse oximeter would actually give a correct reading. Now, the next question is, the lateral spinothalamic tract on the spinal cord carries fine touch and pressure, equilibrium and posture, pain and temperature, vibration and proprioception. Okay, so the tract is spinothalamic, from the spine running, spinal cord running to the thalamus. So this is the ascending tract, and we also know that the ascending tracts, they are carrying the sensation, sensory stimuli, and the ascending tracts are of various kind in our body. Among those various kind, okay, we are basically this various kind belongs to like the one which is coming from the spinal cord, running in the spinal cord, and then ultimately going into the cerebrum. Okay, or the one which is running the spinal cord, carrying the sensation and going into the cerebellum. So now these tracts may either end in the cerebrum or can end in the cerebellum. Okay, so when these sensations are being carried by the receptors present in our body, okay, So various kind of sensory stimuli, they would be detected by their receptors, right? The receptors would now eventually give the information into the afferent neuron, and then this afferent neuron via the posterior gray horn will enter into the posterior horn of the spinal cord, right? Now from the posterior horn of the spinal cord, they will relay the information to another group of the neuron. So whenever they are transferring these sensory stimuli, sensory impulse from one neuron to another neuron, and then whenever they are sending up, okay, they are having they are ascending up in certain anatomical sequence or configuration, due to which the nomenclature has been given, like dorsal column medial lemniscus. That means these tracks, they are running in the white column but dorsally, right? Similarly, some of these tracks, as they are ascending up, they are running in the lateral side, lateral white column, okay. Similarly, if they are ascending up from the anterior side, you call it anterior spinothalamic tract. So these three tracks are actually now conveying the impulses and eventually going into the postcentral gyrus. Similarly, the other tract which is giving the information is also into the cerebellum. These are the from the spine into the cerebellum. So these are the spinocerebellar tract. And remember that cerebellum is associated with the equilibrium and the balancing function. One of its function is cerebellum is related with this function, right? So for doing this function, the cerebellum has to continuously get the information about the position of our body or the joints, and these unconscious proprioceptive kinesthetic sensations are being getting by this cerebellum all the time. So these sensations are being carried from the spinocerebellar tract. And whenever you're talking about this dorsal column medial lemniscus pathway, okay, dorsal column which is running dorsally in the spinal cord, this column is carrying a lot of sensation from the fine touch, tactile localization, tactile discrimination, two-point discrimination, um, and the vibration, pressure. Okay, lot of sensations they have been carried from this dorsal pathway. And if you're talking about this anterior spinothalamic tract, this tract, they are actually carrying the sensation of this group thoughts and tactile localization, and it is via this lateral spinothalamic tract that is responsible for carrying the sensation of pain and temperature. So you can see in this picture that the pain receptors and the temperature receptors, whenever they are being stimulated, these sensations are now going into the posterior gray horn of the spinal cord, right? And then they will relay the information, and this second order neuron will now uh, run along the midline and run into its opposite side of the same segment of the spinal cord, and from there they are ascending up, but in the lateral white column. So this tract is actually the lateral spinothalamic tract, which is responsible for carrying the pain and temperature sensation. So the lateral spinothalamic tract carries pain and temperature sensation. This is our right answer. The next question is, tissue thromboplastin activates which pathway of a coagulation cascade? The options given were intrinsic pathway, extrinsic pathway, common pathway, and fibrinolytic pathway. Okay, so here the question is about coagulation cascade. That means a series of stepwise activation of one clotting factor and another clotting factor, which will eventually lead to the formation of the fibrin thread and stopping the bleeding, right? So if that is the question, the fibrinolytic pathway activation E over here is obviously wrong, right? Now we have to choose among these three options. So let us see. These are our clotting factors. They are altogether 13. So you can see that these are the different names of these clotting factors. You have to know these names as well. Now, these all clotting factors are being activated one after another in a stepwise, ladder-wise pattern, so that eventually the prothrombin would be activated into thrombin, and this thrombin would cause activation of fibrin into the fibrin. That is our common pathway. And this activation of the thrombin can occur via this extrinsic pathway or from the intrinsic pathway. Whenever there's the exposure of blood to the collagen, or whenever there's a change in the constituent of the blood, then there will be activation of this intrinsic pathway. And whenever there is the tissue lysis or blood vessel rupture, at the time the tissue thromboplastin would be released, and they are going to cause activation of factor 7 into its activated form, which will eventually bind, which will eventually now be responsible for this common pathway. So here you can see that the tissue thromboplastin, which is factor number 3, is responsible for the activation of this extrinsic pathway of the coagulation cascade. So with this, we can conclude that the right answer is option B, that is the extrinsic pathway. Now let us move on into the next question. Which parameter will be the same in the pulmonary and systemic circulation? The options given were resistance, flow rate, compliance, and mean pressure. Okay, so to know about these options, which one of them is right, let us briefly recall about our pulmonary circulation. So this pulmonary circulation is actually going to circulate the blood into the lungs. So into the lungs, the blood is actually coming from our right ventricle, right? The deoxygenated blood is coming into our right ventricle, and then from the pulmonary artery, they are going to divide into arterioles and capillaries. From the capillaries, there would be the transfer of the oxygen for the oxygenation of our blood, and then the oxygenated blood would be eventually carried by the pulmonary vein and then drain into our left atrium. Okay, so this system is our pulmonary circulation, and the rest system of our body, from all other aorta to all its branches and veins and venules and draining into the right side of the heart, that is our systemic circulation. So it is very interesting to know that whenever our heart is beating, the right ventricle as well as the left ventricle, both of these ventricles, they are pumping exactly the same amount of blood. But also, the pressure in the systemic circulation is always higher than the pressure in the pulmonary circulation, because the pulmonary circulation vessels, they are having higher compliance. Compliance means distensibility. So if the blood which is propelled out of the right ventricle is of same amount, but it is going into that place that can distend more, obviously the pressure inside this pulmonary circulation would be lesser. So you can also say that the pulmonary circulation is a low pressure system and the systemic circulation is a high pressure system. Okay, so here now we can make out that in the pulmonary circulation the pressure is low, the compliance, it is higher. If the compliance, we say it is higher, that means this distensibility is higher, the blood is easily flowing through it, that means the resistance is less. Resistance towards the blood flow is less. Now, according to our Ohm's formula, we know that the flow is the rate of blood flow is the derivative of pressure divided by resistance. Now, if you talk in the pulmonary circulation, the pressure in the pulmonary circulation is lesser. Resistance is also less. So here, numerator, denominator, both are lesser. That means the flow would be constant. We can say that in the pulmonary circulation, everything would be, which of the following parameter would be same? Okay, the question is, resistance is obviously not same because the resistance in the pulmonary circulation is lesser. The compliance is higher, mean pressure is also lesser, so it is the flow rate which is similar to the systemic circulation. Now let us move into our next question. The synthetic function of liver can be assessed by total serum protein, bilirubin secretion, prothrombin, aspartate transaminase. Okay, so here we are talking about the function of the liver. The function of the liver itself is a very vast topic. There are so many things we have to know about that. And in a very briefly, in this slide, you can see that the principal function of the liver, the formation and secretion of the bile, it helps in metabolism of many substrates, right? Similarly, it is also the site of deactivation or detoxification. They are also helping in synthesis of the plasma protein and immunity via this copper cell. So the question was about the synthetic function. So in the synthetic function, you can see that the liver is the one which is going to produce the acute phase protein, which you can see during the inflammatory condition, the albumin, the clotting factors, and the steroid binding and the hormone binding protein. Okay. Now, among the options which are given were, you can see the total serum protein. So whenever we are talking about the serum protein or the plasma protein, remember there are three types of plasma protein: the albumin, globulin, fibrinogen, okay, and also prothrombin, okay. So among these three plasma protein, the albumin and the fibrinogen is the one which is being synthesized in the liver, okay. So the globulin and other proteins, plasma protein, may not be synthesized in the liver. So total serum protein synthesis is not the function of the liver. Similarly, the bilirubin secretion, bilirubin conjugation happens in the liver, but the synthesis of the bilirubin itself does not happen in the liver. The aspartate transaminase, it can be found in other cells also, not only in the liver. So the correct answer is prothrombin time. Prothrombin time, because the liver is responsible for the synthesis of this protein, that is prothrombin, the clotting factor 2. So if you can remember, we had this formula that the parasympathetic nerves, they have been coming from this 3, 7, 9, 10. And if you're talking about the clotting factors from the liver, they are 2, 7, 9, 10, and also fibrinogen, right? So 2 is the fibrinogen, prothrombin. And by knowing about the prothrombin time, we can actually get the access into the synthetic function of the liver. Okay, so now we'll move into our next question. What is the nature of sympathetic nerve endings in the adrenal medulla? The options given were preganglionic sympathetic, postganglionic sympathetic, preganglionic parasympathetic, and postganglionic parasympathetic. So the hints in the question is already what is the nature of the sympathetic nerve. Now this is the sympathetic nerve. So the options parasympathetic, parasympathetic, both will be wrong. There can be, the options can be between preganglionic sympathetic and postganglionic sympathetic. So let us look into this. So these autonomic nerves, in this picture you can see the difference between the somatic nerves and autonomic nerves. The somatic nerves, they are actually coming from the spinal cord, and then they are going to directly innervate the effector organ. If you're talking about the autonomic nerves, which can be parasympathetic and sympathetic, right? Both of these nerves, before going into the effector organ, they are actually going to relay firstly into the ganglion, okay? And then from the ganglion, the postganglionic nerve will arise, and then this postganglionic nerve is going to affect the effector organ. Now, in case of sympathetic nerve also, similar is the thing that the preganglionic sympathetic nerve is going into the ganglion, where it is going to release the acetylcholine, although it is sympathetic, and the postganglionic sympathetic nerve will now go into the effector organ, where it will eventually release the norepinephrine. Except one of the peculiar feature you can see is the preganglionic sympathetic nerve is directly going into the adrenal medulla, and from the adrenal medulla they would now causing they are stimulating for the release of this catecholamines of the adrenal medulla. Okay, that means this adrenal medulla, they also say that they are actually a kind of postganglionic. They are actually a sympathetic ganglion only, where the postganglionic neurons, okay, sympathetic neurons, they have lost their axon. Okay, so the correct answer would be preganglionic sympathetic neuron is going to supply the adrenal medulla, and in this secretion there will be the secretion of this as a neurotransmitter acetylcholine. So yes, the answer is option A, the preganglionic sympathetic. Now, so let us move into our next question. Which organ is responsible for memory function and control of emotion? The options given were thalamus, basal ganglia, amygdala, and neocortex. So here we are talking about the memory function as well as control of emotion. So when you're talking about memory, it is very important higher intellectual function of our brain, that we are not only learning, but we are capable of storing it and also retrieve it as per our necessity, right? So whenever we are talking about memory, these can be divided into two types. You can see the explicit or declarative type, and the implicit or non-declarative type. So the declarative type of the memory is very specific, very concise, okay, like the facts and events which you can remember, okay, which is very specific. So these are the function of from the medial temporal lobe and hippocampus. And when you're talking about non-declarative or implicit memory, It may not be very much concise or may not be very much specific, but you have the memory about it. Like priming is a process in which you are saying a certain letters, and via that letter now you can remember the whole sentence or word, and this is actually the function of neocortex. Similarly, once you have learn and then repeat and make the habitual of certain procedure like learning bicycle or other thing, okay, procedural thing, once you have learned it and you have acquired that skill, okay, now you can you have that memory to do it, to repeat it again. This is the function of a striatum. Okay, so that is the part of our basal ganglia. Similarly, the other type of non-declarative memory is associative learning, in which there is association with the person can make an association between two stimuli. So this can be classical and operant conditioning. And for the emotional responses, the amygdala here is playing important role, and for the skeletal musculature, this cerebellum is playing important role. Okay, and if it is of non-associative learning, in which there is no association between the stimuli, like in cases of habituation and sensitization, the implicit kind of memory is actually the function of the reflex pathways. Okay, so now with this thing, we can make out that we are talking about the emotion as well as memory. Okay, so that is actually the function of our amygdala, which is part of our limbic system. We all know this thing. Okay, so here the correct answer would be amygdala, because you know that neocortex, it will be associated with the priming thing. The basal ganglia, it would be associated with the procedural memory, right? And the thalamus is actually a group of cluster of the nuclei which is present just above our midbrain, where some of the neurons, they have the specific functions, like they is the relay of certain specific sensory stimuli over it, or they are also forming a part of the Papez circuit, or certain ascending tracts may give the information into it. Okay, and it may also have some role in the motor system. Okay, so the role of this thalamus in the memory can be somewhere around the Papez circuit, but not related with the emotion. So you can omit this thing. So the correct answer is amygdala. Okay. So here with this, now we have concluded the 12 MCQs which came from physiology side in CEE 2077. So the information which I have given you is actually from the standard textbook. Okay, the slides and the sources maybe from the internet, and some of the slides they are from the Ganong 24th edition. And I also request all of the students to please kindly refer to the standard reference book, the standard textbook for the reference. Okay, and with this, I would like to conclude. Thank you and best wishes for your upcoming entrance examination. Namaste.